Okay, so lipids come in a couple of different varieties. We talked about this before. The main ones we're going to talk about are the triglycerides and the fatty acids. So you can see down here at the bottom when we made our list of what these kind of molecules look like. These are the ones that kind of have that long zigzaggy line of fatty acids. And these get attached to a glycerol molecule. And that forms, if three of them attach, we call that a triglyceride. And that's what makes up fats and oils for the most part. But we have one that only has two of these tails, this one up here at the top, and then it's got this phosphorus group, and that's an emulsifier. So that would be in things like egg, so egg lecithin or soy lecithin. And those help mix together things like oils and sugars. So the oils are nonpolar, the sugars are polar, and this has the long tails that will mix well with the oils. And then it's got that phosphorus group with lots of oxygens that will mix well with the lots of oxygens on sugars. Okay, so those are the main things we're going to talk about, but we will spend a little bit of time talking about these steroids over to the side. So cholesterol is the main steroid that we think about. But you also know from, just living in the world that there are other steroids like performance enhancing steroids and things like that. A lot of the hormones that help regulate our bodies, including our sex hormones like progesterone, estrogen, and testosterone, are steroids. So they have this structure that is the four rings that are put together, and then they'll have some things hanging off. So this one is cholesterol. And cholesterol does have important functions in the body. But just like the other lipids, it's mostly nonpolar. So if we think of anything that is fatty or lipidy, we mostly think of those as nonpolar. But this phospholipid here can be an emulsifier. All right, oils and fats. So oils and fats, we said, are mostly triglycerides. They're nonpolar. Nonpolar means it's not going to mix well with water. So another word for nonpolar is hydrophobic. So hydro means water, phobic means fearing. And the main difference between an oil and a fat is at room temperature, what's it doing? So mostly if it's a liquid at room temperature, we call it an oil. And if it's a solid at room temperature, we call it a fat. There are a few that are kind of on the edge there where coconut oil is a solid at room temperature. So if you buy that in the store, it's going to kind of look like a jar of mayonnaise. It looks white. But if you have it in your house on an air conditioned hot day, if it gets into the 90s, that's going to start to clear up and look more like an oil. So its melting point is very close to room temperature. The oil in chocolate, the fat in chocolate, cocoa butter, is also very similar, that it melts very, very close to room temperature. So all fats are about 9 calories per gram. And this compares to our proteins and our sugars are closer to 4 calories per gram. So gram for gram, if you were to eat a pound of fat, you would get almost twice as many calories, no, more than twice as many calories than if you had a pound of sugar. So they are very calorie dense. And this is why we store energy that way, because when we think about, not being protected human beings, but being animals of the planet Earth, animals want to be able to store food for later. And so they have fats as a storage of food. All right, so that's kind of all you need to know about fats for right now. Okay, so we have a couple of different kinds. They have the long fatty acids that hang off of them, and they're nonpolar. Okay, we will come back to them again later. Proteins. Now, proteins are polymers, and rather than just having like 4 parts that we fix together, they have an alphabet. So this molecule on here is the generic form of an amino acid. So we talked about this last Thursday, but as a reminder, when we hear amine, that means we have a nitrogen somewhere. Now, usually it's going to have a couple of hydrogens attached to it, sometimes 3, sometimes 1. But the nitrogen is the key thing that tells us that we're looking for an amino. So that's where the amino part of the amino acid comes from. On the other side of the molecule is this carbon that has two oxygens. So one oxygen has a double bond and one oxygen has an OH. All together, those four atoms are called a carboxylic acid. So that was one of the other functional groups we had to look at. And then the third functional group was an alcohol. So one of those is going to show up on your quiz today. So I'll review them again after we go through this slideshow. All right, so we see this R here. The H is just a hydrogen. The R, remember, can be lots of different things. So I had the model where I had one of the amino acids just had another hydrogen and another one had a couple of carbons attached to it. There are some that have rings. There are some that have additional nitrogens on them. So there are some that are non-polar if there are a bunch of carbons, and there are some that are polar if they happen to have more oxygens and nitrogens on them. And so because of that, proteins are also emulsifiers because they have some parts that are going to be non-polar and some parts that are polar. All right, and so we see in this picture here listed the hydrogen, the amine, the carboxylic acid, and the R is just the rest. There are about 20 that make up our proteins. And then there's another handful that the amino acid exists biologically, but maybe isn't part of a protein. There's a real important one we're going to be talking about later in the semester called GABA that is in your brain. And so being able to use food to regulate that GABA can be helpful. But we'll get back to that. All right, so just decided to include a cartoon here. What do you call an acid with an attitude? It's an amino acid. So I mentioned that there are about 20 that our body uses. Of those, 11 of them we can kind of make from raw materials. We have enzymes and cell functions and things that will do that. But there are 9 of them that, unless we have something that's almost identical to the starting material, we really can't make them. And so we call these essential amino acids. You don't need to know which ones are essential, but you need to know that an essential amino acid is one that we have to get through food, that we can't make it in our bodies. All right, so we talked about emulsifiers. Remember, the emulsifier has one part that's polar, that's going to mix well with water, and one part that is non-polar, and that's going to mix well with oils. So our proteins work as emulsifiers because they have the amine and the carboxylic acid parts are polar, and then the carbon side chains are going to be non-polar. And then they have some of the side chains that have nitrogen or oxygen, and then some of them have sulfur. Those tend to be a little bit more polar, especially the ones with oxygen. Now, those parts that are non-polar tend to be a lot smaller. They're not long, floppy chains of 18 or 20 carbons the way that we have in fatty acids. So rather than trapping oils, these are much better at trapping things like air molecules. So air molecules can get trapped up in the proteins. So if you've ever had a meringue, like a meringue topping on a lemon meringue pie, or there's the little cookies that look like a Hershey Kiss, but they're like practically all air. That's just an egg white, which is mostly protein, that has been whipped up to trap the air molecules. Okay, so the non-polar parts trap the air molecules. The polar parts have the water, which is what makes the egg white runny. And then as you whip it up, then you are getting some of that water to dissipate out. And then as you cook it, the rest of the water, most of the rest of the water. All right, so proteins serve as emulsifiers because they have pockets that are polar and pockets that are not polar. So now we have 2 emulsifiers that we've talked about. We have our phospholipid, and then we also have our proteins. So while amino acids have 2 polar functional group, the side chain can be a non-polar one. This means that proteins tend to be, this is a, were you just paying attention moments ago question. All right, so these tend to be emulsifiers is the correct answer there. Polypeptides. So the bond between two amino acids is called a peptide bond. So a polypeptide is just several of these amino acids linked together. So sometimes you will hear that term polypeptide, that they might use it in a commercial about medicine or your digestive system or whatever. So each amino acid that makes up the proteins in our body is assigned a letter of the alphabet, because there's only 20 of them and we have 26 letters. So we have 20 letters that we use. And so this very much makes it like we're spelling out a word. Now, a polypeptide is just a handful of proteins, but when we make a handful of amino acids, so it's just a short word of maybe 5 to 20 letters. But a protein, remember, can be hundreds to thousands of amino acids long. So that would be a really, really, really long word. All right, so the monomer, the smallest piece is the amino acid. We build the words to be polypeptides, but the really long polymer is the protein. All right, proteins have 4 levels of structure. The first level is just the order, the spelling of the word. And then depending on how the polar and non-polar parts fit together, we can get this secondary structure. This is where it just starts to kink and bend in its own local area. So one of these is a helix. So you can kind of see this twisting around. That's called a helix, or an alpha helix. And the other one is called a beta sheet. They just kind of fold back and forth like an accordion. Not too concerned about identifying the secondary structures, but when you see a picture of a protein, because the molecules are so big, you're often going to see the helices or the beta sheets represented. So when you see pictures of corkscrews and folded things, you should be thinking proteins. That's just showing you the secondary structure. So this would be like the zoomed in, looking at just a very small part of the protein. So we have primary, secondary. The next level is called tertiary. That's right, I can do it with the arrow. Click. The tertiary structure is when you look at the whole protein, from the beginning letter to the very last letter of the whole protein, and it's all folded up together. So the secondary and tertiary structure are really kind of related to one another. So this is, rather than looking at just one little corner of the protein, it's looking at the whole big protein. And then proteins can get together in groups to do things. And one of the ones you've probably heard of is hemoglobin. So hemoglobin is a glob, the globin part of 4 different proteins put together. And that's what we call our quaternary structure, is when we have 4 of them put together. So you should know that we have 4 layers, right? You should know that the first level, the primary level, is just the order of the amino acids. And you should know that the third level, the tertiary level, is the whole big protein shape. Those are the 2 important ones. All right, I said before that the fats are about 9 calories per grams. Proteins are between 3 and 4 calories per gram. Sometimes we estimate right around 3.5 calories per gram. So we think of these a lot as being in meat, but of course, everybody needs proteins, including vegans and vegetarians. And so there are a lot of plants that have proteins. So beans and nuts are really good sources of protein. Grains can be, but it depends on which part of the grain that you are eating. So if we think of wheat as a grain and we buy wheat flour, well, most of the wheat flour that we buy, the white flour, has been stripped of pretty much everything but the starch and a little bit of protein. Whereas if we have the whole grain, then we've got more vitamins, fats, proteins, all of that. So we should know that it can be found in both plants and animals, and what plants in particular would be good for that. They also control the expression of the genes. So your DNA, the whole point of your DNA is to tell your body to make proteins. And so that means that your DNA, which is what tells you who you are, is making proteins that go out and do the work of making you who you are. So when you express a gene, when your eyes are brown, when your hair is red, there is some protein out there that is working to make those things happen. Sometimes we have sort of a negative gene, a recessive side of that, which is I'm not expressing that. So lighter colored pigmentation is just I'm not making as much melanin or whatever is coloring your eyes or hair or whatever. But all of that is controlled by proteins. So they do a lot of different things. I said we express like our hair color and things like that. The amino acids and shorter peptide chains make up some of the hormones that run our bodies. Not all of them, because remember, some of them were those cholesterols, but there are things that that are more protein shaped than they are cholesterol shaped. Some of the antibodies in our body, muscles, of course. So all of those are by proteins. Proteins are super, super important. Now, one of the most important proteins is called an enzyme. And this is where regulation comes in. So an enzyme is just a special kind of protein. And I think that's on the next slide, but I still see a couple of people writing, so I was going to wait a second. The enzyme is what we call a catalyst. So you may have had a science class before where you've talked about catalysts. Its basic job is to speed something up. So we have lots of molecules in our bodies that need to hit each other in just the right way in order to react. And they're really big, complicated molecules. So if we just left this up to them sloshing around in solution and running into each other, it would take years for those reactions to occur. So what enzymes do is they hang on to one part of a molecule so that another molecule can come in and attack it. Or they hang on to two molecules real close by so that they can get together. Okay, so they speed up the reaction by giving those molecules that need to react a place to react. So some of the ones you may have heard of are lactase. This one's real important to me because I am lactose intolerant, but I love dairy food. So anytime I go and I have ice cream, I take some lactase. So enzyme names, you'll notice the ending here is ACE or ASE. That's pretty common. Lactase helps to break down lactose. Now, most, if not all, mammals are born with lactase being made in their gut because mammals drink milk from their mothers. Every mammal has had milk, and that breaks it down. But humans are kind of unique in that we continue to drink milk throughout our lives or dairy products throughout our lives. At least some people. Some people don't like dairy products. And so while nature would sort of have us stop needing to have lactase after we, sort of reach middle childhood, humans continue to consume dairy. One of the things that we get from milk is vitamin D. And that's really important for helping to incorporate calcium into our bones. Now, our bodies can also make vitamin D if there is a lot of sunlight around. So if you think geographically, places that are further north are places where dairy tends to be more important in the diet. Scandinavian countries, Wisconsin, they love their cheese, that kind of thing. Dairy tends to be less important in the diets of people as you move closer to the equator. Now, as the world has become more global, a lot of that has changed. But if you look historically over thousands of years, that's kind of what we're looking at, is that people in northern countries typically had more dairy in their diet. And so people who continued to consume milk would need to continue making lactase in their diet. And so lactose intolerance is less common for people whose ancestors are from Scandinavia, northern Canada, those kinds of things, than it is for people who are whose ancestors are from closer to the equator. Now, that's not 100%. Anybody can be lactose intolerant. Anybody can tolerate the lactose, depending on where you're from. But the general trend tends to be people whose ancestors are from northern countries tend to be more lactose tolerant. Amylase is an enzyme. So remember, we're talking proteins here. This is a protein that breaks down amylose, and this was one of the starches we talked about. So carbohydrates have starches in them as one of their components. And so it helps break that down into the simple sugar. So this long chain of sugar molecules stuck together can't get from your gut into the rest of your body. And so amylase, starting in your mouth, starts breaking that down into the smaller bits. And then there's other processes throughout. And then tyrosinase is another one that we'll talk later on about. But this one is responsible for making fruit turn brown. So if you've had an apple or a banana sit out too long and it starts to turn brown, like after you've cut it, that's tyrosinase that's doing that. All right, so enzymes do a lot of different things. Those are just some examples. They are essential for food growth. They help things to ripen. And they also lead to spoilage. So there's lots of different things there. All right, so another, were you just paying attention moments ago question? What role do enzymes play in chemical reactions? All right, they speed up biological reactions. Excellent. They are catalysts. So yeah, all of these that refer to catalysts, special proteins, all of those are excellent. So let's see. These are ones that I would have said, but these other ones are also correct as well. So people are paying attention. Excellent. Okay. One of the things that we do a lot when we're cooking is to destroy proteins. Now, we don't make them not be proteins anymore, but we change their form in such a way that they can't go back. This is called denaturing and coagulating. So remember, they have that tertiary structure where they're all kind of balled up in a particular way. Denaturing allows us to pull them apart so that they're more in their long string shape, and to coagulate. And so that we take one protein and kind of join it to another protein because it has these parts that weren't exposed before, and now they are. They can join forces there. And so there's basically three ways to do it, three stresses that you can do to a protein. So if we think of an egg, we can do these all to the egg white. So the egg white is water and protein. We can heat it up. Okay, so I had a fried egg this morning for breakfast. It went from this clear goo to a white solid. Okay, now when I cooled it back down to room temperature, well, I didn't get it down to room temperature, but it cooled down before it went into my mouth. It didn't turn back into that clear goo. This is not reversible. Okay, so those proteins were unfolded and then kind of clumped together. This is why we have fevers. As our temperature rises, even just a few degrees, it changes the shapes of the proteins in our bodies, which is going to change the way that any bacteria or viruses might try to attach to our cells. And their proteins are also changing shape. And so it's hoping to change them just enough that those bacteria either are killed or at least cannot infect us as easily. We think of the average body temperature as being what? So 98.6 in Fahrenheit, or does anybody know in Celsius? 37 in Celsius, okay? So this was done by looking at records in the 1800s for hundreds and hundreds of people. And really what the person that was looking at this was looking for was what happened when people stayed at the hospital longer. Did they get a fever that went up, stayed up, and went down? And what he found was it kind of went up and down, which your parents probably found when you were little, was that you would have a fever and you'd be like, oh no, and then you call the doctor and go to the doctor and it's like, this kid doesn't have a fever. And mom's like, I swear they had a fever before we left this morning. And that's what happens. It goes up and down. So they took averages of healthy people and they found that it was about 98.6. Really what they found was that it was about 37 Celsius. That's two significant figures. But when you change that to Fahrenheit, it becomes 98.6. We really don't know with that much precision. There's a range. People have plus or minus 0.3-ish going in either direction. What has happened over time, though, is that as we have become better at keeping things clean and being in healthy environments and those kinds of things, our body doesn't want to have to work so hard to keep us warm. And our bodies have learned, you know, if I only go to 98 degrees or 97.8 degrees, I don't have to work as hard, but this person seems to stay pretty much as healthy. And so if we were to do an average of healthy people right now, it would probably be closer to 98 than to 90.6. The average healthy temperature is going down. On the other hand, bacteria, viruses, funguses are learning and adapting to a warming climate. And so they're getting better at surviving at warmer temperatures. So where it used to be that this is where the bad stuff was and this is where we are, we're kind of getting closer together. And so this makes some of the illnesses that are coming out in the news a little bit harder to treat because our own ability to fight against them with our normal body temperature is going down. Okay, so heat is one way that we can denature a protein. Another way is with chemicals. And so acids is the way we normally do this. If you have ever seen pickled eggs, what they do with a pickled egg is rather than hard boiling it, they put it in an acid like pickle juice or vinegar or something like that. And the acid is what changes that egg white to white instead of clear. So that's another way that we can denature a protein. And then the last way is by physically stressing it. So we talked about meringue before. If you're whipping the proteins, that's going to trap some of that air in there, but it is also denaturing, unfolding the proteins and whatnot so they don't just collapse back down. So beating eggs, whipping eggs, those also denature proteins. Which molecule, so this is kind of thinking back of all of our biomolecules that we've looked at, right? We've had simple sugars, we've had amino acids, we've had cholesterol, we've had proteins. Which of these is the largest and most complex? Majority have this correct. So the protein was the only one where we talked about having like 20 different little pieces that we could put together in lots of different ways. Okay. So that makes it very complex. And then because there are hundreds of them rather than just 4, that makes it very large. So our proteins are very, very big. And then all of the parts are different. And so that makes it complex. All right. I think we have one more question. Which functional group is found on proteins? The only one that we found on proteins, but not on the other biomolecules. All right, so this one we had a little bit more trouble with. So the amine is the one that has nitrogen, and that's in proteins, which we call the amino acids. Okay, so good one to know there. Proteins have amines. Your sugars are basically in rings. And then hanging off of the rings, there are a bunch of these OH groups, carbon OH. So sugars have which functional group? Do you remember what the carbon OH was? This is the alcohol group. And we're going to see alcohols a lot this semester. So that one's probably the most important one to remember. And then our fatty acids, that acid name comes from the carboxylic acid. So we have this long chain of a whole bunch of carbons. And at the end, there's a carbon that has 2 oxygens attached. One oxygen is double bonded, and one oxygen kind of looks like the alcohol, but the whole thing put together, that is called a carboxylic acid. Okay, so those are our functional groups. We are going to jump into chapter 3 on Thursday. So your vocabulary for chapter 3 is due tomorrow night. So don't forget that. Chapter 3, we're going to talk about measuring things. We're also going to talk about crystals. On Tuesday, we will have our exam over chapter 1.